Hello, everyone, and welcome to today's webinar titled COVID-19 Testing, Results and Recommendations from the Coronavirus Standards Working Group. This webinar is part of the 8th Coronavirus Virtual Event Series, and I am Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by LGC Clinical Diagnostics. For more information about LGC Clinical Diagnostics, go to lgcclinicaldiagnostics.com. So let's get started. Before we begin, I want to remind everyone that this event is interactive, and we encourage you to first participate by communicating with other attendees using our new live chat feature during the presentation. You can find that live chat located at the left of your screen. You can also participate by submitting as many questions as you want during this presentation. To do so, simply type them into the Ask a Question box and click Submit. You will also notice that this speaker has provided a survey located at the right of the abstract. This survey will be available during the live session and during the on-demand period. Now, if you have any trouble seeing or hearing this presentation, just click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby. And finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on that continuing education credits link located at the abstract window below the presentation window and follow the process to obtain those credits. I now present today's speakers, Dr. Marc Salit, the Director of the Joint Initiative for Metrology in Biology at Stanford University, and Dr. Eric Morreale, the Director of Product Management at LGC Clinical Diagnostics. For a complete biography of our speakers, please visit the Presenter tab from your menu at the left of your screen. Dr. Salit and Dr. Morreale, welcome to both of you. You may now begin your presentation, gentlemen.
Hi, thanks so much for that nice introduction, and I'll reintroduce myself as Marc Salit. I'm a MITRE Fellow and a adjunct professor in the bioengineering and pathology departments at Stanford University. And this is a little story of what I did in my pandemic. And I'm happy to be presenting at the LabRoots event here on the idea behind and the work products of the Coronavirus Standards Working Group. We created a global forum for talking about and meeting on and trying to figure out some of the nuance of the infrastructure for COVID testing. And I'll give you a little background on this. I'm going to move to the first real slide here. Back in March of 2020, we were all shutting down. I was canceling events and canceling trips and canceling work programs and trying to figure out what my unique capabilities could bring to what had clearly emerged as a global problem. And I'm a standards guy. I make stuff and put it in tubes and give it to another scientist and tell them what's in it. And I often say that's the most audacious thing a scientist can do, is hand another scientist a tube of something and tell them what's in it. And all of a sudden it came to this place of, okay, what are we doing for standards and calibration materials and quality system support for COVID testing? And so I started ultimate grassroots, making calls and writing emails to people I knew around the world who were clinical chemistry laboratorians, as well as people in professional societies and government agencies, to find out who had this. I would call somebody at a federal agency and say, do you guys have this? And I was not hearing that it was managed and everybody was drinking as fast as they could from the fire hose. But this seemed like a gap. And so we formed up this effort, and you can see on the right panel, this is a slide I made for our very first sort of town hall meeting, which was held on March 27th of 2020, about what a coronavirus standards working group might do around assuring the tooling was available for successful rollout and scaling of what we were still calling at that point 2019 novel coronavirus, eventually became COVID, or SARS-CoV-2, and what infrastructure was going to have to be in place in order that we had a COVID testing enterprise that we could trust and that would function at scale. So the working group, I called out that we should identify and develop the critical infrastructure to support confidence in test results, interoperability of results from different systems and different tools and different labs, scale up and long-term capacity. My goodness, I look at this slide now, which is October of 2022, and I wish we had all this still. So we certainly fell short on influencing the world in the most powerful ways. But I think we did a lot towards this third bullet or third little box, identifying the best practices that should be institutionalized. And we established a global forum and a global network in doing so. The history I was just enumerating here, I initiated this at a program that is the Joint Initiative for Metrology and Biology, joined at that time by the National Institute of Standards and Technology. It's part of the U.S. Department of Commerce. We established weekly meetings very quickly, and we ran about 60 of those weekly meetings overall through the duration so far. And our cadence dropped to a couple times a month, and then our cadence has been intermittent as we've been trying to get some products out the door and publications out, and people have transitioned into different roles, myself included. I was then director at JIMBY, and now I'm a fellow at MITRE Corporation, which is a nonprofit organization in support of public safety and well-being. We had broad participation from the ecosystem, and that's one of the things, and I'm going to foreshadow my concluding slide, which is a thank you. One of the things that made this all work and happen was that we had participation from the clinicians who were overwhelmed at the moment, the professional societies like the ASM and like CAP and the federal agencies like the CDC and the FDA, so public health authorities and public health academic efforts, as well as the technology developers who are making test equipment and test reagents, as well as the standard developers and the innovators throughout the field. People in the global metrology community, people in the effort to do interlab testing and virology, really amazing convening that we were able to host. I'm not going to even say we convened, we hosted it, and it happened. And throughout, there's been terrific situational awareness sharing and knowledge sharing around best practices and emergence of new capabilities, as well as, wow, did you see that this organization has made available standards of this sort, and this is how our experience with them, or this is how they can best be used. It was really a terrific forum. And we took on one very concrete study that I'll talk about in a couple moments, to do harmonization to the international standard that was developed by the National Institute of Biological Standards and Controls on behalf of the WHO. And that's the effort in that's the institute in the UK that does a lot of development of the WHO standards. So our work program at this point looks like a paper that is now in press. It's recently accepted at Nature Biotechnology that talks about a roadmap for better COVID testing and reliable public health information through having standards infrastructure. And we also, in that roadmap paper, make reference to a draft of a terms of reference for an emerging infectious disease standards working group. We make the recommendation that such a working group be established and be a standing capability for the globe. We also then conducted a harmonization study that was 14 labs, 8 different materials, calibrating those 8 different materials against the WHO international standard that by that time had been fully established and adopted. Those standards were of inactivated virus, recombinant virus, and recombinant bacteriophage makeup. And I'll get back to more of the study design, and you'll see the variety of different labs that were also involved. But first, I'm going to talk about this roadmap paper. Oops, we lost the title of the paper in this slide. The paper is, though, called A Roadmap to Better Testing. And it is in press and will be in the November 2022 issue of Nature Biotechnology. You can see there the list of authors. And it's a terrific list, ranging from national laboratories to universities, to people from that National Institute of Biological Standards and Controls, to commercial testing laboratories and national measurement institutes, as well as a panoply of academic contributors. One of the things this paper does right out of the gate is discusses testing as a measurement process. Oh, wait a minute, here we go, sorry. This was the work program discussion. I think I did what the facilitators recommended against in terms of advancing my slides, but rewinding a little bit, this was the work program for the roadmap paper and harmonization study. This is, in fact, the roadmap paper author list. And this was the first activity in the roadmap paper, is identifying that testing really is a measurement process, and there's roles for, so the measurement process is outlined on the top part of the slide, and there are roles for different types of standards to do assurance of quality and content of the test results, and the different kinds of controls and standards that we identify throughout the test, throughout the roadmap manuscript. As I noted, there were recommendations about a standing emerging infectious disease working group, infectious disease standards working group. And key things to note: independent, international, standing network of members that then would know each other when need might emerge for development of infrastructure to have a reliable test enterprise. And the roles would be to establish those relationships, develop and communicate policy and advocate, and provide representatives to organizations who are in fact trying to manage response. The details of this, as I say, are in the paper that will be in the November issue of Nature Biotechnology. Let's go to the next slide. I mentioned above, and I have to zoom back up here, that we had this working, this harmonization study as a work program element with 14 labs, 8 different standards evaluated of different categories. And the purpose of the study was to establish the equivalence of target concentrations of different materials in this panel against the WHO International Standard Reference. It was not to test labs. Going back here, it was not to test labs and evaluate quality of labs. It was not to test or evaluate quality of standards. It was merely to value assign this panel of reference standards. This was the design concept. It's close to the actual design we accomplished. There were 4, in our case, inactivated virus samples. There were I believe 2 recombinant virus and 1 recombinant bacteriophage. I'm sorry, 2 recombinant bacteriophage. So we had a total of 8 samples in the panel and a dilution series of the WHO international standard, which at that point was no longer the candidate standard, but what had been adopted. And the providers of all the different standards contributed their materials to this effort, as did the WHO contribute the calibration materials. These went to clinical labs, test developers, and national measurement institutes, each measuring the full panel and reporting results. And we had a portfolio of testing labs that used quantitative PCR and some that used droplet digital PCR. So we had a real nice panoply of data, and are currently writing the manuscript that did the value assignment. We have an open public dashboard where all these data are available, unembargoed, and it's available on a platform that was developed within the working group in the R Studio Shiny app ecosystem. And here's a snapshot of what that looked like. And in this case, you're looking at a calibration curve on the left panel of this snapshot. And that was from an organization called Biodesics for the N1 target. That's their calibration against the WHO reference material and their droplet digital PCR count. And there were, as I say, 14 labs, and most of the labs had at least 2 targets. And you can see the results on a sample. This was the NIBSC sample that was an engineered virus calibration material that was also released from NIBSC. And that's the box plot on the right. Here's another snapshot of that. This is a different lab and a different target. This was Los Alamos National Labs clinical laboratory, the calibration on the left. And this on the right is the LGC Sericare engineered virus calibration material and the value assignment, where the y-axis of that is the concentration, and the lab's results are plotted as boxes and rank order. Open data, open analysis, and there's the link to the app that has this full dashboard. And I'm sure that LabRoots will make that available as a clickable link along with this presentation. On the right of this panel is the summary results from all 14 labs and all the targets in IU per mil for the 8 different materials in the study: Assuragen, Imperial College, Instand, LGC Sericare, Microbiologics, NIBSC, Thermo Fisher's Acrometrics material, and Septometrics. And those have a 95% confidence interval associated with them. And those are not rank ordered. Those are just alphabetized from A to Z. An interesting graph is on the right. What is on the right here is the results that were putative based upon the nominal copies per microliter of the international unit and the reported copies per microliter plotted against the results in international units. So the fact that most of the materials have a confidence interval that crosses this best fit line is heartening. And that best fit line, in fact, is consistent with the nominal value in copies per microliter of the international unit. This is the sort of take-home shot from this study, and as I say, the manuscript's in preparation at this point. The manuscript is being championed and first authored by Sarah Suleiman at the UC San Francisco Immunology Department. Another side effect of this work was colleague and I, Tim Mercer, who's at University of Queensland in Australia, wrote a review on application of any kind of SARS-CoV-2 testing to a population scale. And this review made the cover of Nature Reviews Genetics back in July of 2021. And Tim and I are sort of bringing the band back together to work on this harmonization study manuscript with Sarah. And in fact, Tim is the first author on the roadmap paper that's due to be published in November in Nature Biotech. Oh, and I think we've seen that slide change. There's the slide change. Sorry, I'm button impaired apparently today. There's the note regarding Tim's and my paper and Nature Reviews Genetics from July 21. I'm going to sum up at this point with a huge acknowledgement of everyone who contributed to the Coronavirus Standards Working Group. We had a couple of hundred people on our mailing list, and attendees at our weekly meetings probably ranged up to 300 over the series of meetings, with a core probably of about 50 who were regularly attending, especially as we were undertaking this harmonization study. I'm excited that this work, just to put this in context, really has never been attempted before in terms of dissemination of an international unit in the time of a global pandemic in order to have comparable and trustworthy test results from clinical measurements. And this I think stands as an example of what kind of capability the scientific community has in the 2020s in response to global needs. And I hope we've established some best practices, and I stand behind the recommendation we make in the roadmap paper that a standing emerging infectious disease standards working group be established to continue the preparedness of response to global disease. And again, with that, I thank you, and I know I'm turning it over now to a colleague at LGC. Cheers.
Thanks, Mark, for that great overview of the coronavirus standards working group. I think it's really important work. So I'm glad we're getting to see the fruits of that labor. Again, just a quick introduction on my end. I'm Marc Morreale. I serve as Director of Product Management at LGC Clinical Diagnostics, really focused on commercialization of molecular controls, and was really leading the effort, certainly during the early days of the pandemic, to develop controls that could be used globally by clinical labs to ensure accuracy of their test results. I did want to provide a little bit of an overview on the technology we utilize to develop the molecular controls that were actually used in the coronavirus standard working group study. It's based on what we call a proprietary Accuplex technology. And what's unique about this technology is we're able to mimic a true RNA virus in the case of SARS, but design in a way that it's completely safe and non-infectious. So you can see it's a little bit of an eye chart on the left, but essentially the way this works is we're able to discretely identify a target sequence of interest, and combining that with helper RNA, transfect a cell line that generates virions that are genetically engineered to be replication deficient. So this enables development of material that's safe, and certainly in the case of SARS in the early days, it was hard to get access to material, and there were a lot of questions around safety. So we're able to bypass some of the challenges in the beginning of the pandemic, particularly in getting access to true viral material to make controls that were mimicking SARS-CoV-2, but also had the features of being safe and, importantly, fully extractable. So one of the challenges, obviously, with just generating, although it's quick and easy, naked RNA as a control material, the problem is it doesn't really evaluate in the extraction step of an assay. And so our product, using Agiplex technology, not only is non-infectious and safe to handle, but it does require extraction. So you're evaluating that full process of the assay. In addition, it mimics a patient sample. We're able to put in multiple matrices depending on the particular virus we're looking at. We developed it in both kind of a VTM type matrix as well as on swabs, and it's a completely independent third-party control. Another benefit of this technology is it's highly stable. So typically we'll get 2 to 8 degrees storage for up to 2 years, which certainly when you're working with either a real virus or just naked RNA, you don't get that kind of stability. So there can be cold chain storage challenges with utilizing that. And then, as I said, it's completely customizable. So for SARS, due to the multitude of assays that are on the market, many targeting different sequences, we developed a full genome SARS-CoV-2 product in that case, but it is customizable. So we can target up to, in a particular Agiplex, 4 kb length of RNA or DNA, and really target a specific assay as needed, or consensus sequences. And so historically, we've used this ideally when you're dealing with pandemic materials or pandemic viruses like SARS-CoV-2, but also viruses that are kind of nasty, and you don't want to be working with the real thing like Ebola and norovirus. Also have developed multiplex products using this technology. The most recent are SARS-CoV-2 flu and RSV products. And so that's kind of the technology we used to enable us to get control material that was very badly needed in the early part of the pandemic for labs to be able to validate assays. And there were a lot of issues with assays, certainly back in March of 2020, to be able to validate that those assays were performing properly. And then just quickly to close out, just to make people aware, our intention is based on the results and the work that Marc and his group did with the coronavirus standards, is to develop an Agiplex quantitative panel where we're actually converting the dPCR counts, digital PCR counts, to the WHO IU calibration. So our intention is to develop a product to be able to really verify assays across the assay range, past the upper range, and also below the LOD. And so I think, utilizing this technology, we can expand on the toolbox that we have, and certainly utilize the technology to be prepared for future pandemics. And so with that, I want to thank everybody for attending, and happy to take questions.
Thank you, Dr. Salit and Dr. Morreale, for your time today and for your important research. Before we go, I want to thank our audience for joining us today as well. And I'd like to thank our sponsors, LGC Clinical Diagnostics, for sponsoring today's webinar. Questions submitted today and during our on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand for 2 years until October 19th, 2024. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's. Everyone, and bye-bye.